All right. It's 3:30. Let's get started. Thanks everyone for being here today. I have the honor of introducing you to Dr. Peter Balint-Kurti. He's a research geneticist with the USDA Plant Science Research Unit at North Carolina State University. He attended Cambridge University as an undergraduate and received his PhD at the Sainsbury Laboratory, housed at the John Innes Center in the UK. He's held postdoctoral appointments at the NIH and the Boyce Thompson Institute at Cornell, and worked at 2 biotechnology companies before joining the USDA in 2003. Since joining the ARS, his work focuses on elucidating the genetics controlling quantitative disease resistance and the defense response in maize. Today, he'll be speaking to us on a number of his research projects in his presentation titled Trying Not to Overreact, Mechanisms Controlling the Maize Defense Response. With that, I'm pleased to hand it over to you.
Well, thank you. Thank you very much for the invitation. Thanks, Becca. Thank you. Thank you for coming to hear my seminar. It's a great pleasure to be at Purdue again. I've been here a few times. And as Becca mentioned, I'm a USDA scientist. So I'm an ARS scientist, and I know you have a bunch of those in your department as well. I'm stationed at NC State University. I've been doing this for more than 20 years now. I've been in this position for more than 20 years. And today I thought, rather than just talk about my latest projects, this was an opportunity for me. I've given talks here before. I wanted to sort of go back through some of our research projects and try and sort of pick out a theme that you might be sort of illuminating. I don't. Well, you tell me if it is. But so the theme that I chose is trying not to overreact. How maize? I work on maize disease resistance. So think about how maize controls the defense response. You need to be resistant to plants. You can go to the next slide here. Most plants are resistant to most diseases. Most plants have defense mechanisms, and those mechanisms come at a cost. And you can make this sort of fairly obvious analogy to a walled city where the city, back in the times when it was built, very well defended, but that defense comes at a cost. It costs resources to build these walls. It costs resources to surveil them, to man these lookout points and look for enemies that are coming. That's all takes time and resources. That you also have the issue of you lose the ability to easily exit and enter the town. So these are all costs. They're associated with an effective resistance mechanism, and same thing goes for plants. Same thing goes for us, actually. And there's a trade-off. There's a trade-off between defense and growth. This is well known. I didn't discover this, but I wanted today to discuss some of our research projects in this context. Research that sort of pertains to this growth defense trade-off, that illustrates its effects, that illustrates some of its consequences, and also some of the mechanisms as well that I think seem to be controlling this trade-off. So we're going to go through several projects, and at a fairly shallow level, I'm afraid. I hope they're understandable, but I'd be happy. Stop me if they're not. But I also wanted to introduce some of the main protagonists today. The diseases we're looking at. Southern leaf blight is a disease that I've worked on for the longest. It's a foliofungal pathogen. We also work on another foliofungal pathogen, grey leaf spot. So southern leaf blight is caused by this fungus, Cochleoblus heteroscopus, and I'm going to mention that name again. Grey leaf spot caused by a different fungus. And northern leaf blight, another foliofungal pathogen. These are all relatively necrotrophic diseases caused by related pathogens, actually. And really, almost all my research is really asked the question: what's causing variation in resistance? What are the genes? What are the loci? What are the genes? What are the mechanisms involved in controlling sort of naturally occurring variation, standard variation in resistance. So when you think about, at least when we in the MPMI field, molecular plant microbe interactions field, think about recognition. There are different types of disease resistance you can think about, but one of them, the most commonly sort of discussed, is recognition-based immunity. What I mean by that is the plant cell actually recognizes the presence of the pathogen and then induces a defense response. There's two sort of main types of this. If you think about plant cell coming into intimate contact with a pathogen cell, so two broad categories of molecules that the plant can use to recognize the presence of the pathogen. One set of these molecules are called PAMPs, pathogen-associated molecular patterns, and these are molecules that are, well, that can be shed from the pathogen. Flagellin is sort of the archetypal PAMP. Most of you have probably seen discussion of that. But they're not these. Often these molecules are relatively highly conserved, and they're not specifically associated with pathogenesis. They're more associated with general microbial lifestyle. So flagellin, for instance, required for movement, and that's not. I mean, it's required for pathogenesis, because the bacteria have to move in the plant cell, but it's also required just for everything else. So PAMPs are produced in the apoplast, and some of these PAMPs, like flagellin, are recognized by pattern recognition receptors, and often pattern recognition receptors, or actually all the time, they work in concert with co-receptors. And there's a defense response, there's some sort of signal transduction, doesn't matter what it is, for our purposes, there's a defense response. And that's called PTI, pattern-triggered immunity. So in general, I mean, there's different ways of defining PTI, but here we're defining it as a defense response that's initiated by recognition outside the cell. And then there's a class of molecules called effectors, which are molecules that are somehow introduced into the plant cell by the pathogen. These molecules, usually proteins, are generally associated with pathogenesis, and they do all kinds of things. Doesn't matter what they do for our purposes. But some of these effectors are recognized by resistance proteins inside the cell. And they're recognized. Actually, what happens then is this resistance protein undergoes some rather extreme structural changes and oligomerization. Again, it doesn't matter for our purposes. There's a recognition event. There's a defense response. And this is called effector-triggered immunity. So for our purposes, effector-triggered immunity is an immune response that's triggered inside the cell. And if you have PTI and ETI together, they potentiate each other, at least we think they do, at least in Arabidopsis. And you get a bigger defense response. This defense response often includes a reaction called the hypersensitive response, which is a rapid localized cell death at the point of pathogen penetration. And often that's, the relationship between HR and resistance is a little murky, actually, but often that's sufficient to confer resistance. This is a picture of arachnosis. This is HR at a single cell level. We see it in maize in my system of choice. So here we have a maize line that is susceptible to common rust. Here is a maize line that's resistant to common rust because it has this resistant gene RP1D, and these spots on the leaf, they're not disease symptoms, they're a hypersensitive response. They're the consequence of this ETI plus PTI loss. So when you have this type of resistance, the sort of yes or no resistance, all or nothing, at least I could term that qualitative resistance. So you have resistant or susceptible. Usually this is based on the segregation of single genes of presence or absence of single genes, as is the case in the previous slide with RP1D. And when these genes are segregating in a population, the population usually is just a binodal. There's a binodal distribution here. stuff that's resistant, stuff that's susceptible, not clean, or should be nothing in between. So you can really tell, you can determine the genotype of the plant by its phenotype. And this can be contrasted with the other type of resistance we work on, which is quantitative disease resistance. That's disease resistance that's where the phenotype is distributed in a more continuous way. And actually, this is a scoring sheet I give to people when they score stuff in the field. So we just score it by eye, actually, but so 9 is very resistant, 2 is very susceptible, and then actually 1 is dead. I don't show 1, but it's just a brown leaf. And then there's lots of stuff in between. And this kind of resistance is based on the action, typically it's based on the action of multiple genes, each of which has a small effect. So when these genes are segregating in the population, you often get the population varies sort of continuously. Often you get some sort of normal type of distribution. So we're gonna talk a little bit about quantitative resistance, and then we're going to come back to the recognition next week. So when you think about quantitative resistance, I talked about some of the genes that are involved in this qualitative resistance, but the quantitative resistance, is any gene that has any effect on the pathogen, the symptoms that you see on the plant. Any gene that is whose presence or absence, or any allele whose presence or absence has an effect on the level of the symptom you see on the plant is in fact a quantitative disease resistance gene. So think about sort of archetypal pathogenesis, sort of progress. In our case, we're working with fungal polypathogens, and often the spore will be in the air, either blown by wind or rain, blowing around, lands on a leaf. It has to stick to that leaf, has to germinate on that leaf. That spore has to germinate on that leaf. It has to get into the leaf, has to get through the cuticle or the epidermis, or maybe through the stomate. Often you have to deal with the microbiome, sometimes maybe trichomes, sometimes there's some preformed substances on the leaf, in the leaf. Any genes in the plant that cause variation in some of these things that will affect these stages of pathogenesis could be a resistance gene. So, for example, a gene that affects leaf angle may affect how many spores can land on a leaf. Like you might imagine that more spores would land on a flat leaf than on a vertical leaf, maybe. Anything that affects the stickiness of the leaf, maybe. Anything that affects the stomatal density, if the pathogen has to get in through stomate. Anything that affects the opening or closing of stomate, maybe trichomes, maybe something that affects the microbiome, maybe something that affects the thickness of the epidermis or the cuticle, something that affects the level of these preformed antimicrobial compounds. Any genes which have alleles, 2 different alleles that affect these traits in different ways could end up being detected as quantitative resistance genes. And then, of course, once you get penetration, you get to this point where you have this intimate contact with the plant, and then you have this recognition-based resistance response. Again, there's a lot of genes in this pathway that could end up being quantitative and conferring quantitative levels of disease resistance. So we've done an awful lot of mapping for quantitative disease resistance genes, and please don't read all our papers because they're all the same, right? Just basically like this. This is one of our better ones. So here we used a 5,000 line population. It's called the noun population, and we mapped southern leaf blight resistance limits. And these are, you've probably seen these type of Manhattan plots here. So these are all of these points of molecular markers. They're SNP markers. So we have the number of chromosome 1 to 10, has 10 chromosomes. So here they are on the x-axis. Here's the sort of probability that any one of these molecular markers is associated with variation in resistance to southern leaf blight. So you have some of these markers that are highly associated with variation in resistance to southern leaf blight. And in some cases, I've indicated the name of a gene above the marker, and that's the gene that is closest or usually in which that marker resides. So these genes are the most likely genes to be the cause of the cause of this variation, the causal genes. But they're not getting from candidate gene. So these are candidate genes to actually identifying the causal gene is probably a lot of you know not a straightforward process. We've done this for a few of the genes. I'm not going to talk about any of them except for this last one, this leucine rich repeat LRR protein kinase. So we cloned this gene, or at least we identified it as the causal gene underlying the variation in southern leaf light at that locus. And it's a leucine rich receptor kinase. We call it CHSK1. That stands for Cochleobus heterostrophes, which is the causal agent of southern leaf light susceptibility kinase 1. So it's a gene that actually causes susceptibility to southern leaf light. You can see that here. This is a wild type part with 2 functional copies of that gene. Here's the mutant part. These are near isogenic, so they're almost entirely identical genetically, except this guy has 2 mutated copies of CHSK1. Much more southern leaf light resistant. You can see there is some southern leaf light. So that's quantitative resistance, but it's less. There's much less. So that's interesting. It's a susceptibility gene. Or you could call it a recessive resistance gene. And we looked at this gene. What we're looking at here is actually the PTI response. We took PTI is the response that's triggered outside the cell. And here, this is the mutant line. It's a mutant for the CHSK1. And this is the wild type line. So mutant, wild type. What we're looking at here is this is effectively production of reactive oxygen in species when induced with FLAG22, which is an epitope of flagellin. So we could let's just say it's flagellin. We're adding flagellin to leaf pieces of the mutant line or the wild type line, and then looking at the PTI response as measured by production of reactive oxygen species. And you can see the mutant line reacts a lot more strongly. So stronger PTI response, more resistance when you don't have the functional copy of CHSK1. So what's going on? What we think is going on, and we haven't proved it, is that we think CHSK1. It looks like. I mean, I said oh well, I'm not sure I said this already, but it's a leucine rich repeat receptor kinase, and those type of genes are commonly PRRs. They're commonly pattern recognition receptors. They're the kind of things that recognize patterns, but also they also are commonly co-receptors as well. So so in this case, we're showing, this is a figure that shows FLS2, which is the flagellin receptor in the cell membrane, and it's there together with BAC1, and both of these things are LRR receptor-like kinases. So BAC1 is a I guess a activator receptor kinase. It's required for the transmission of the signal after recognition into the cell. So it's BAC1 is a sort of an activated co-receptor. But then there are also these genes or these proteins that are inhibitory co-receptors. So in this case, BF3 is also a co-receptor, but it's an inhibitory co-receptor, and actually it can also interact with BAC1 to remove it from FLS2 and inhibit the activation of PTI after flagellin recognition. So we think CHSK1 may be acting as an inhibitory co-receptor. That's one of our theories. So to get back to sort of the causes of quantitative disease resistance, which I suggested were many and varied, I think this is borne out by the 4 genes we've identified as being associated with quantitative disease resistance. We think we have an inhibitory cell surface co-receptor. We've cloned a gene involved in the MAP kinase cascade, which is the signal transduction system that because after PTI, after the initial PTI recognition, we've cloned a gene involved in nidification, and one involved in amino acid transport. And you can see all of these genes, these 4 genes we identified, they seem to be doing very different things, but they all, and on the cellular, on the molecular level, but they're all conferring quantitative levels of disease resistance. And obviously, we've cloned 4 of these genes. If you look at the literature, there's now hundreds of quantitative disease resistance genes identified, and there's all kinds of mechanisms associated with them, and we've actually written a couple of reviews on this. The 2 of these, I've already mentioned this CHSK1, but also this guy, this amino acid transport gene, they are recessive resistance genes. In other words, their functional copy confers susceptibility. And we've been interested in them. Recessive resistance genes probably, maybe they don't get the respect they deserve. Often when you do mapping experiments, and again, please don't read my mapping papers, but often you don't really look at recessiveness dominance because you're dealing with inbred lines. That's the easiest way to do the mapping. but often I guess I would always assume that disease resistant genes would be dominant or semi-dominant or partially dominant. But actually, when we look at it, we've done some recent studies where, some of them are semi-dominant, but then some of them are dominant, and some of them are recessive, and it's about equal numbers of recessive and dominant quantitative resistance genes. So these recessive resistance genes are quite common, and I think probably more common than we appreciate. Recessive alleles generally imply loss of function, and so as I mentioned, a recessive resistance gene implies the absence of a gene confers resistance. So the presence of a gene confers susceptibility. So how does that work? Well, there's two basic possibilities, I think, and probably both pertain. So one is that the gene may encode function needed by the pathogen. And you can imagine the pathogens have been co-evolving with the host for a long time. And so, they've adapted to exploit certain host processes. And there's certainly a lot of evidence in some cases that pathogens exploit the functions of certain host genes. So when you take those away, the pathogens, its mechanism of pathogenesis is compromised because it's evolved to target particular genes. Now, I'm not going to talk about that today. But the other possibility is the gene may be inhibiting the defense response. It actually may be suppressing the defense response. When you take it away, you have a heightened defense response. You have more resistance. And that, I think, is what we're seeing with CHSK1. So then the question is, well, why on earth would you evolve this whole defense response system, this whole disease resistance system, and then evolve a bunch of other genes to suppress it? I think we know the answer to that. You can analogize that to the car. Cars we built them to get us to places, but you need to be able to stop them as well. You need to be able to control where you're going, control how fast you're going. Otherwise, the problem, you can't, you overreact. That's bad for growth. You can think about us as well. It's like if you run too high a fever, often a lot of the symptoms associated with COVID, for instance, was more of an overreaction to the pathogen than caused by the pathogen itself. And there's a lot of autoimmune diseases and diseases that where the symptoms are actually caused by overreaction. You need a break. So one of the most well-known recessive resistant genes is MLO, and I'm not going to go into too much detail, but it's in barley. It causes resistance to barley powdery mildew, and when you don't have it, so it's a recessive resistant gene. When you don't have it, which is here, it's associated with a certain amount of yield loss, a certain amount of abiotic spotting, so flecking. And we see that same thing in maize. It is B73, which is a commonly used maize line. That doesn't have much flecking, but you can see some of these other lines, they have this sort of spotting, this mild spotting. And we were interested in whether some of the spotting was sort of analogous to this MLO, whether the spotting was maybe a low-level defense response, and whether it was associated with disease resistance, and also with yield penalty. So they're generally recessive. Maize, and the reason I was interested in is because I talked to some maize breeders, and they said, sometimes they think that lines which show spotting are good sources of resistance. They incorporate them into their programs preferentially because they think they're good sources of resistance. So we wanted to look at this in a systematic way. What we did, and it's a little bit complicated, but what we did is we took this 5,000 line population. I already talked about where we did the mapping of the southern leaf line. We scored each line for spotting. So, and we scored on the 0 to 6 scale, I guess. And then we sort of thinned them to this being low spotting, and the spotting, the flecking, as we called it, gets more extreme. Go this way on the x-axis. And then we looked at a few phenotypes of each of these groups. We looked at yield, and that's shown on this dotted line. I think that's purple. You can see as the flecking gets higher, you have less yield in these lines. And then we looked at disease resistance. Let's look at northern leaf blight. And here we show this is decreasing yield, but the same direction is actually showing increased disease resistance. So you have increased disease at high flecking group, increased resistance, decreased yield. And then for the other two pathogens, we look at southern leaf blight and grey leaf spot. You see some increase in disease resistance again with increased flecking. So you see, so that's you're seeing a correlation of flecking, increased resistance, decreased yield. So you're seeing evidence of this growth defense trade-off. Less growth, less yield in this case, more resistance. In this paper, we suggested that at a level of mild flecking, which is sort of somewhere here, you're seeing you're maintaining yield, but you are seeing some increase in disease resistance. So maybe mild flecking is a nice marker for some breeders to use for selecting lines. And there's better markers than that, but it's an easily scoreable marker. So that's flecking. What about quantitative disease resistance genes themselves? Is the presence of a quantitative disease resistance allele associated with changing yield? Does that depend upon the presence of the disease? We asked this question a while ago, and this was a lot of work. What we did was so we evaluated the resistance and yield effects of the two recessive resistance genes I already talked about, this amino acid transporter and the CHSK1 inhibitory co-receptor. What we did was we created a bunch of hybrid lines, and we used hybrids because maize is generally, maize is growing as a hybrid, and yield is really only relevant in the context of being in a hybrid. So we created isohybrid lines, and what I mean by that is the identical hybrid, except one was homozygous for the presence of the resistance allele, one was homozygous for the presence of the susceptibility allele, which was wild type in this case. And then we actually created a bunch of these pairs, and then we grew them in side by side in inoculated fields, so fields inoculated with southern leaf blight, and then the field right next door, in fact, the same field, we just split it up, that was not inoculated, furthermore was fungicide treated, and the idea there is to stop the spread of the pathogen from the inoculated field. The idea, having them in pairs like this is obviously we're trying to minimize the effects of the field environment. And we did that over multiple environments over multiple years. And to cut a long story short, here are the major results. We have no disease. The hybrid, which is basically the line with the susceptibility allele. It grows a little bit better because you get a little bit better yield than the hybrid with the CHSK1 or the hybrid with this amino acid transporter resistance allele, resistance to. So if the hybrids have the resistance, this quantitative resistance allele, both of them, when there's no disease present, less yield. When there is disease present, more yield. The regular hybrids, you get some decrease in yield because of the disease, not as much as we were expecting actually. So, and then the yield is protected in the lines with the resistance allele to some extent. So there's yield penalty in the absence of disease, as a yield protection in the presence of disease associated with the quantitative resistance. We've just looked at 2 genes, and that was a lot of work. Is this generally true? I don't know. I assume, I assume it is to some extent for some genes. I assume this is the case, but really there's been no other studies like this that I know of. But I think we can assume that at least some parts of quantitative resistance are associated with decreases in yield. And that then begs the question, that suggests the question, what happens if you develop lines evolve in the absence of disease pressure? Do they lose these resistance alleles? And we were able to ask that question, at least in an indirect way, recently. We used a population developed by Ruben Relan Alvarez, who is a colleague at NC State. And this is a little bit complicated again. But basically, we have about 300 lines that all share a common genetic background, but have about 25% of their genome sort of randomly derived from land races, derived from ancestral land races that were developed all over South, Central, and North America. And then we assessed them for Southern America. We did that all together in North Carolina. So this is this map shows to each of these spots, one of those lines, one of those 300 lines, placed at the point where its landrace progenitor was derived from. If that makes sense. So, and then the color corresponds to how resistant it was to southern, right? So blue in this case is more resistant, red is less resistant. And we can here's the center of approximately the center of maize domestication, southern Mexico, the lowlands of southern Mexico. And from there, about 10, well, I guess it was developed 10,000 years ago, and then the subsequent years, maize has moved both north and south. And you can kind of see here that as it's main blue in the area of original domestication, and then as it's moved north and south, it seems to have, in general, there's more red. So Asher Hudson, who did this work, he took all the environmental variables he could associated with all the different locations, where our land race is derived from, and he tried to find associations between environmental variables and disease resistance. And the 2 top associations were with temperature and rainfall. So land races that were developed in areas with high rainfall and high temperatures were more resistant, conferred more resistance on their progeny. Land races that came from dry, cold places, more susceptible. And that was true for both North America and South America. You can see, so this is cold, dry, warm, wet, cold, dry, warm. So it looks like maize, in both cases, maize populations lost resistance as they moved north and south from warmer, wetter environments to colder, drier environments. And as you can probably guess, southern leaf blight does best in these warm, wet environments. So the idea is, if you develop maize, develop land races in these colder environments, where southern leaf blight is less of a problem, some of these quantitative resistance genes are selected against, and you lose them sort of preferentially, and sort of loss of resistance, again, implies a cost to resistance. Again, we have this growth defense trade-off in evidence. And you can think about this. I often think about. I've had aquariums since I was a little kid, and you can buy these fish in aquarium shops. They're blind cave fish, and they evolve in caves. And there's no light in caves, and they lost their eyes because eyes are expensive to make. And if you don't need them, if there's no light, you don't need them. Why have them? These just they kind of swim around, they bump into stuff. And so the same thing with resistance. If you don't need it, and it's costly to maintain, then you're going to lose it during evolution. So we talked about quantitative resistance. We've talked a little bit about flecking. I just wanted to end by talking about recognition-based resistance. So I talked about PTI and ETI recognition outside the cell, PTI recognition inside the cell, ETI. And both of these things are costly in terms of growth as well. And PTI actually is that cost is very easy to see. Actually, both are very easy to see. So here's an example where we've looked at the PTI response to flag 22 again, to flagellum, in a bunch of different maize lines. And just basically look at this, you look at these black bars. CML228 has a strong response, B73 much lower response. And if you grow, you can actually grow maize or little maize seedlings in flagellum. So you kind of you're overstimulating this response, and you can see that when you do that, you go B73 in 10 micromolar flagellum, and this is the check, so just in water basically. Not much difference. CML228, which shows a much stronger response, growth is really compromised. So you're activating this PTI at a very high level, admittedly. You're having a profound growth, a profound effect on growth. So again, it's growth, defense response in this case. Trademark. And we were interested in this because we think this is a little bit of an aside, but we think, I was just talking to Damon about this, how everybody has their own explanation of heterosis, and your explanation depends on what you work with. So if you work on disease, you think disease explains heterosis. So here's my attempt. This is work that Maggie Wagner did when she was in my lab and is still doing. So heterosis, most of you know, it's the phenomenon whereby you cross two inbred lines, the hybrid, the F1, is much more vigorous in terms of growth. The yield is much higher. And here we're looking at two very commonly used maize lines. I've already talked about B73, most 17 is also very commonly used. And they produce a very vigorous hybrid which yields very well. So Maggie looked at growth of these two inbreds and the hybrid in the presence in sterile conditions, or when she added a synthetic bacterial microbial community. What you saw, so here's the F1 in the middle here, it's two parents with the symptom, with this synthetic community. You see sort of heterosis. You see more, in this case, this is root fresh weight. You see better growth of the hybrid than two inbreds. But if you remove that synthetic community, you grow them under sterile conditions, not much heterosis. In fact, and that seems to occur because the inbreds are doing worse, not because the hybrid is doing better. And our thesis, at least one of our theses, we had a few, but one of our theses was that hybrids may manage this growth defense trade-off better than inbreds. And we have some evidence for that. So here we've looked at the PTI response again, same way, added flagellum, looked at reactive oxygen species production. So here is most 17 actually responds quite well. B73 doesn't respond that well, but it does respond. Here's the hybrid green. It's in between the two parents, which is what you'd expect with sort of additive genetic model, although it is closer to the lower parent. Okay, that was I actually did that sort of when he came in 2 hours after dawn, which is 2 hours after the lights came on in the growth chamber. He did it again. Initially, he did it again for totally unrelated reasons, but he did it again 4 hours after, so 2 hours later. And what you see is quite remarkable. And he's repeated this several times since, but again, Mo-17 is a higher responder. B-73 does respond, but much lower level. But look at the hybrid. No response at all. So there's a circadian element here, which we haven't quite worked out. But what we're seeing is the hybrid is responding at a much lower level than either of its parents, at least at this time. And yet we also have evidence, it's actually, the hybrids are very resistant. So it seems like it's suppressing its immune response while somehow maintaining and actually improving its resistance. So somehow we think the hybrid is better able to control, better able to deal with this defense growth trade-off. And maybe that's an element of heterosis. I wouldn't say it explains heterosis entirely. And actually, I will say these type of experiments are very difficult to reproduce. These data are reproducible, but not all of our data in this project are. That's what I said already. So that's PTI. What about ETI? I just want to mention this sort of classic experiment. This was done in Joy Bergelson's lab many years ago, 20 years ago. What they did was they created isogenic pairs with and without a resistant gene RPM1. So they did this in a clever way. I won't go into it. Basically, they created lines that were entirely genetically identical to the nucleotide, except one had RPM1 and one didn't. And they created several of these pairs, which were identical. So one had RPM1, one didn't have RPM1. In every case, in every pair, the line without RPM1 yielded more seed. So there was a yield penalty carrying RPM1, which is a resistance gene, Pseudomonas syringium. So what's going on? There's a 9% thickness class, and that's crazy because this is not the only resistance gene in Arabidopsis. Actually, Arabidopsis has more than 100 of these type of genes. So 100 times by 9% is 900%. So that each of these genes can't be associated with that level of thickness loss, but this one was. And we think it's probably because these resistance genes, they need to be able to detect the pathogen very quickly. They need to be able, as soon as they recognize the effect, they need to be able to turn on. So they're very sensitive, and a very sensitive kind of switch. And you can imagine these very sensitive switches, like a fuse switch or something. Sometimes they get triggered inappropriately. So I think that's what's happening with RPM1. It's being triggered occasionally at some level inappropriately, and you're getting this growth penalty due to that. So that's in Arabidopsis. In maize, I already showed you RP1D, that confers resistance to common rust. Now we have an allele of RP1D called RP1D21, and we actually did this work. This work started here actually with Yuri Howard. We worked with him for a long time on this. But RP1D21, the presence of RP1D21 causes this spotting, and there's no pathogen here. This is just a spontaneous hypersensitive response. So I want you to notice two things here. One is the spotting caused by RP1D21, and then also that you have different levels of spotting depending on the genetic background. So each of these lines has RP1D21, but each has a different genetic background. So what we see is genetic background effects. So there's alleles here which are enhancing this HR. There's alleles in this line which are suppressing it. You have that kind of situation. You can map these genes, you can clone these genes. It's not easy, and we spent a long time doing it. Oh, I just wanted to mention, so here, for example, is a line that's segregating for the presence of RP1D21, wild type. So there you have the growth defense trade-off again, this is a very exaggerated form of defense response, but you can see if you initiate ETI, if you stimulate ETI too much, you do have quite a growth effect. Some of them actually have even worse than that, some of them depending on the genetic background. So we mapped a bunch of modifiers, again using this 5,000 line population. Again, it was a lot of work. We've had a lot of, these are candidate genes at this point, but we've cloned a lot of these genes. We actually identified 45 loci associated with variation in HR caused by RP1D21. Now I'll come back to that. We identified several of these genes. Some of them are involved in ubiquitination, which is a system that marks proteins for degradation in the cell. And I'm going to talk about one of them, which is the ZM-surnine E3 ligase. And E3 ligases are the proteins that specify which cells are marked for degradation. They specify that they directly ubiquitination the marking of specific proteins, usually for degradation, sometimes for unequivalent. So here we're not using maize here, obviously we're using Benthamiana relative to tobacco. And we can express RP1D21 in this system, this is transient expression, and you get cell death, which is what you would expect. You co-express it with GUS as just a sort of negative control, still get cell death. Co-express it with ZM-9, no cell death, or very little cell death. So it looks like, you know, ZM-9 is the gene underlying variation in RP1D21 conferred HR at that rate. And ZM-9 is, as I said, an E3 ligase. We also looked at the physical interaction between ZM-9 and RP1D21. And we've actually, these are a bunch of different RP1D alleles. And the two I've marked as yellow, those are autoactive alleles. And the two autoactive alleles interact with SIR9. That's shown by the growth in this yeast-2 hybrid, the growth of yeast in its yeast-2 hybrid assay. Their growth on these plates without histidine indicates interaction of these two proteins. And then we have three other alleles which are very similar, very, very similar. Here's the wild type RP1D, and there's a couple of other ones which are non-autoactive. None of them are associated with SIR9 physically. And we did this with yeast-2 hybrid. We've also done this with another technique called bifluorescence complementation. And we get the same result. So we have this E3 ligase. It seems to be interacting specifically with autoactive NLRs. And is it causing degradation of these NLRs? We think it is. So here's RP1D. It's a non-autoactive NLR in the Western blot. Here you co-express it with GUS. Again, no real change in abundance. Co-express it with ZM-9. No real change in abundance. RP1D21, which is the autoactive NLR. Co-express it with ZM-9, much less protein. So we think ZM-9 is actually causing the degradation of the autoactive allele, the activated form of the protein. Not only that, I guess I'm not going to go over this in interest of time. I'm not really going to go over this, except to say that there's ZM-9 has a homolog, an ortholog in Arabidopsis. If it's doing the same thing in Arabidopsis, when you knock it out, so we've already showed that the ZM-9 suppresses the HR, suppresses HR. So when you knock it out in Arabidopsis, you would expect more HR if it's doing the same thing. And that is what we're seeing. Here we have two lines where we've knocked out the gene, and we've induced HR using a pathogen here, two different resistance genes actually, in a completely different system. But you see HR in the wild type, but at sort of normal levels. In the mutants, without SIR9, we see more HR, and we can show that quantitatively also. So what do we think is happening here? What do I think is happening here? Our model is that, I mentioned that these resistance genes, when they're activated by recognizing an effector, they undergo these profound structural changes. They actually oligomerize. They form a calcium channel, and they insert into the plasmid membrane. So I think that this SIR9 protein, I think it's recognizing the activated forms of these NLRs, and it's targeting them for degradation. So we think what's happening is This represents a resistance gene. I don't think I mentioned it. That NLR stands for nucleotide binding loosely rich repeat protein. So this is a so-called CCNLR. So it's a very typical type of resistance gene. When it recognizes the presence of an effector, of its cognate effector, its structure changes a lot. It initiates ETI, initiates HR, but then also ZM9 recognizes it, ubiquitinates it, targets it for degradation. So this, this activated form doesn't last very long in the cell membrane. It's targeted for degradation and it's soon degraded. So this may be, I think this is a general mechanism for suppressing the immune response after it's activated. So, you need this HR needs to be activated very quickly. And then it needs to be shut off. When it's done its job, it needs to be shut off very quickly. So I think that's what's going on. I think that gene is one of the genes responsible for it, and we think it's a general mechanism that is present in maize, is present in Arabidopsis, and may affect all resistance proteins of this type. So just in conclusion, I hope I've demonstrated some of the ways we've associated disease resistance with reduced growth. Quantitative resistance mechanisms seem to reduce growth, at least some of them. Flecking, which we think is a marker for sort of increased disease defense response level, that reduces growth, enhances resistance. And then recognition-based mechanisms also seem to conform to this defense growth trade-off. If over evolution time under reduced disease pressure, you can imagine that some resistance mechanisms may be lost because of selection for higher yield, selection for higher reproductive fitness. And there are clearly implications for breeding there. I think breeders already know this. If you're a breeder, you need to select your plants under conditions that are relevant for the field, and that includes diseases. You need to select your plants under the relevant disease pressures. But if you don't, you end up losing resistance, and actually that happens all the time. You work in maize, you're aware that we have some cyclical disease problems which occur when the disease really hasn't been a problem for a long time, and maybe breeders haven't really selected for resistance to it, and then suddenly it becomes a problem again because the germplasm, the germplasm has just not been selected for resistance and it's lost those resistance values. Talk about heterosis and maybe how some part of heterosis may be due to the fact that hybrids can manage this growth defense trade-off better. We don't really understand why that is, or we don't understand why that is. And talk about the genetic architecture of ETI, controlling ETI is complex. RP1D21, there's at least 45 loci that affect its activity. 45. That's at least 45 genes. It's very important to keep resistance proteins inactive until they're acquired, until they're needed. And there are consequences if you don't. And then we've elucidated at least two of the mechanisms we think that control the defense response. One is the CHSK1 gene, which is an inhibitory co-receptor, we think. The one is CM9, which we think degrades activated resistance protein. And then just wanted to sort of leave you with the thought that there's RP1D21 is one gene, but there's 45 genes that are controlling its activity. Furthermore, there are more than 100 there's 150 resistance proteins in maize. So, if there's 45 genes controlling each of those, that's, I don't know, I haven't done the math, but it's a lot. And clearly some of these modulating proteins are affecting more than one NLR. You can imagine there are a lot. I mean, maize has 150 of these genes. Each one of these genes has a potential very easily to kill a cell. So it has to be controlled very carefully. So you have 150 sort of loaded guns, as it were, in the genome. And you have all these genes that are controlling their expression, all these proteins that are controlling their expression. That is a lot of the cell's energy going to produce and manage the defense response pathway, the defense response there's a lot of. Just, my point is that defense response, disease resistance is very important. In fact, sexual recombination is believed to have evolved as a way to deal with pathogens, as a way to modulate, to recombine our genomes to evolve with pathogens. But anyway, disease resistance is important. I'll leave you with that thought. So I just want to acknowledge the people involved in this work. Shannon and Greg, Shannon Summers and Greg Marshall are two long-term technicians who really are the engine of our lab. They do all our planting, all our field stuff, and Shannon is our lab manager. And with that, they really are the key to any success we've had. I talked about work done mainly by Wendy Liu and Asher Hudson, and I showed pictures of them on the appropriate slides. Then I also have several other very talented people in my lab whose work I haven't been able to talk about. And then I've talked about the work of some previous members of the lab, including these folks. And then all of these folks are very important, or have been very important collaborators to whom I'm grateful, and I'm also grateful to our funders, NC State, USDA, NSF, mainly, but there are some other funders as well. Thank you.
Does anyone have any questions? We have about 5 minutes.
Well, I mean, we don't know. I don't really. I think what I think is we see these lines of going flecking. Probably there's multiple mechanisms that are associated with why we see the spotting. But some of them we think, we have evidence that at least some of them are associated with the defense response. So, my guess is that some of the flecking is maybe mild form of observation. You mean like? No, not that I mean, I think I know what you're getting at, but not that I know of. No, we only looked at those 3 pathogens, but those 3 are largely necrotrophic pathogens. You're thinking that rainforest susceptible to necrotrophic pathogens, not which part of the. But really, we don't know them. It's hard to get to the mechanisms we have. I think this because it's a recessive trait and it's a mild trait. It's very hard to even that. So we have it. And your core structure. Yeah. Yeah, that's the other possibility. Yeah. So you're suggesting that it's actually a resistance gene that's exploited by necrotrophs. It's causing HR. That's a possibility, and that why there's a reason that I think that's coming on. Well, one thing is, so we have actually looked at that, and we've done those experiments, the same type of experiments that Paris and Friesen did, where we take a sort of fungal extract and we infiltrate it to leaves, and we looked for necrosis caused by fungal extract. We found a differential necrosis, but that does not map to CHS where CHS1 is. So we have no evidence for that. But I mean, that is a possibility too.
I'm just curious, if the CHS1 is this kindness, it makes sense. It says recessive. But what about the amino acid transporter? How kind of recessive amino acid transporter in resistant swing for chocolate acid? They're all oxo chocolate.
Well, the short answer is I have no idea. But it's interesting that same gene has been associated with resistance in arachnoids. And there they talk about control of reactive oxygen species. And I don't really understand how they get that, to be honest.
Under a microscope, whether the fungus can penetrate, are they just
under the microscope? I mean, basically, the actual fungus can actually get into the leaf, and then it just kind of stops And I don't, yeah, that's all, yeah,
It's got traffic, I mean, they kill it. But if they're friendly.
it, you know, it can't, it gets in and it can't get much further. That's all I can say. It's actually, this is actually a well-known southern leaf blight resistance locus. It's called the RHM1. I don't know if you've heard of that, but yeah, I don't know the mechanism of that either. Yes, we have some more work to do. What you're suggesting? I think that is true.
So I was interested in Asher's data showing the flag, the response to flag claims. Do you ever do that with a different sample listing? For example?
I don't think we've done it with chitin. What we have done it with different hybrids. We've done it in different
Other
genetic
than
backgrounds.
resin.
Yeah, and then we use other parent combinations, and we basically do the same thing. I don't think he's done it with chitin. I don't think he's done it. Yeah, I mean, right. You know, I'll acknowledge, and I think we discussed this today already, that you know, we're measuring PTI in we're measuring one action of PTI, measuring elicitation. In this, what I showed you is one elicitor, one output measure. There's lots of different outputs you can measure. There's lots of different listers you can use. And, you know, we actually have experience where you get very different answers if you use different listers. So, you know, this is a nice story. Yeah, but yes, it's a story.
Alright, well, you have a yeah. Oh, yeah, that's a student.
So thank you so much for your presentation. My question is regarding about the disease pressure and also the looking the different races that this
Well, that's a good question. The answer is we do our inoculations with sort of a mixture of different aspects. We don't have races in the same way as you might be familiar with a lot of biotropic pathogens. You know, there's different races that overcome different resistance genes. If you look at the nectar example, you don't really have major resistance genes for this pathogen. We do. There is a race called race T, which you may have heard of in your plant pathology classes.